Frederick is a biochemist who has made many contributions in the field of molecular biology. His contributions have had a lasting impact on genetics, biotechnology, and medical research. He has won the Nobel Prize in Chemistry twice. Among victories are the discoveries related to the kinase of amino acid together in the protein insulin and the determination of the sequence of bases in nucleic acid. Frederick's first victory to the work produced. His achievement in insulin sequestration was that he terminated the complete amino acid sequestration of insulin by developing technique called protein sequestration to determine the sequestration of amino acid in protein. Before the creation of this technique, the actual sequestration of amino acid in protein was difficult to know, and after Frederick Singer developed this technique, the actual sequestration of acid will be identified. Next, Frederick Singer received his second victory together with two another biochemists named Paul Berg and Walter Gilbert. For this technique, he has determined the sequence of base in nucleic acid, and he has used method to decipher genetic material. Berg was taking with basic research on the biochemistry of nucleic acid and recombinant DNA. Gilbert determined the sequence of bases in DNA, while Frederick Singer led to the determinant of the structure of another complex protein, stimulating research on protein structure and enabling chemistry. The three of them were not only that, the resulting technique is known as the Sanger technique, after her name Frederick Singer.
Next, method of determining the base sequence of nucleic acid, Singer sequence. Singer sequencing, also known as the chain termination method. Singer sequencing involves three main steps. First, DNA sequence for chain termination PCR. Modified nucleotide called ddNTPs are added to stop DNA extension, resulting in terminated DNA strand of varying length. Manual sequencing set up for reaction with a single ddNTP type each, while automated sequencing uses all ddNTP in one reaction with unique fluorescence label for each dNTP. Next, size separation by gel electrophoresis. Size separation of chain terminated oligonucleotide is done through gel electrophoresis, where DNA fragments are placed on a gel matrix and pulled toward the positive electrode by an electric current. The speed of movement is based on size, with smaller fragments moving faster. Lastly, gel analysis and determination of DNA sequence. DNA polymerase synthesis DNA from fifth to third direction, with each terminal ddNTP corresponding to a specific nucleotide. Manual Singer sequencing involves user interpretation, while automated Singer sequencing uses fluorescence and a computer for identification, resulting in a chromatogram.
Next, let's move to the application of nucleic acid-based sequencing. The first application is the identification of genetic variations and mutations. These applications applies in studies of mutations and heritable disease. It is also able to define a tumor's mutational spectrum and identify a constitutional variant in diagnostic testing. For instance, when a kid has an autosomal recessive illness like cystic fibrosis, this Sanger sequencing allows for predictive genomic testing in at-risk relatives, such for a familiar BRCAI variation conveying breast cancer risk and carrier screenings for parents. Next is the determination VNTR or variable number tandem repeat sequences for crime scene profiling or paternity tests. This application is widely used in field of forensic science for criminal cases. VNTRs or variable number tandem repeat are an important source of RFLP genetic markers used in linkage analysis, which is the mapping of genomes, which uses polymerase chain reaction (PCR) that determined by gel electrophoresis and certain blotting to produce a pattern of bands unique to each individual. The first person to be convicted in criminal case on the basis of DNA evidence in the UK was Robert Milius. Also, in the same year in the US, Tommy Lee Andrews was convicted in a rape case based on DNA evidence in which his DNA profile was matched with that of semen traces recovered from the victim.
Application of nucleic acid-based sequencing. HLA typing. Human leukocytic antigen HLA typing is a genetic test used to match patient and donor for bone marrow, cord blood, or organ transplant. The HLA system composed of gene and proteins from the major histopathology complex MHC, aiding the immune system in identifying body cells. Genotypic microsatellite markers. Genotypic microsatellite markers analyze genetic variation in population revealing highly polymetric variation in STRs, repetitive DNA sequences with one anti-base pairs. Microsatellite genotyping is the identification of the genotype of each microsatellite, a repetitive DNA sequence found in thousands of locations within an organism's genome. These sequences, which are repeat multi-times, contribute to genetic diversity. The process involves prime design, PCR amplification, and polymerase testing. Microsatellite have various applications including forensic and medical fingerprinting, cancer diagnosis, genetic link analysis, and population genetics.
Okay, now let's moving on to the advantages and disadvantages determination of base sequence of nucleic acid. Starting from the advantages, firstly, low sequencing errors, which mean this method has low error rate compared to other sequencing methods available at the time. Secondly, this method produces long contiguous sequences of DNA, which beneficial for assembling larger genomes and identifying genetic mutation. For the third one, it provides high accuracy in reading DNA sequences. And the last one, it gives detailed genetic information that allows for the precise determination of the exact sequence of nucleotides in DNA molecule.
Next is the disadvantage of base sequence of nucleic acids. First is high cost, which is sequencing nucleic acid can be expensive. Sequencing nucleic acid, especially for whole genomes, can be expensive. Despite reductions in cost over recent years, comprehensive sequencing projects still require substantial financial resources for reagents, equipment, and computational analysis. Number 2 is complexity and technical challenge. The process of sequencing and subsequent data analysis involve complex methodologies that require specialized skills and knowledge. Errors can occur during sample preparation, sequencing, and data interpretation. Additionally, maintaining high accuracy in sequencing results can be challenging, particularly with homopolyramic regions or repetitive sequence. The next one is data management and storage. Sequencing projects generate vast amounts of data that require significant computational resources for storage and processing. Managing this data, ensuring its security, and making it accessible for analysis can be logistically demanding. The last one is bioinformatics challenge. Analyzing sequencing data requires sophisticated bioinformatic tools and pipelines. Researchers need to handle issues related to sequence alignment, error corrections, and the identification of variants. This requires expertise in both biological science and computational methods.
Discussion on Frederick Sanger's DNA sequencing method. Number 1. Preparation of DNA template. The DNA that needs to be sequenced is separated into single strand. A primer, a short single-stranded DNA fragment is annealed to the template strand at its 3 ends. Number 2. Reaction setup. We prepare 4 reaction mixtures, each containing DNA polymerase, all 4 and one of the 4 labeled with a tag. Number 3 is DNA synthesis. DNA polymerase extends the primer by adding complementary to the template strand. Occasionally, may be incorporated instead of leading to termination of DNA strand. This results in DNA fragment of varying length, each ending where the was incorporated. Next is electrophoresis. The reaction mixtures are loaded onto separate lanes of a polycarbonate gel and subject to electrophoresis to separate DNA fragments by size. The gel is then exposed to X-ray film for radioactive labeling or detect using a fluorescence scanner for fluorescently labeled. And lastly, sequence threading. The position of the terminated fragments indicate the DNA sequence. By reading the sequences of bands from the gel, the DNA sequence can be determined from the shortest to the longest fragment. Application of Sanger sequencing. Genetic research used to sequence specific genes or regions of interest to identify mutations associated with genetic disorder. Fundamental in the early stage of the Human Genome Project, providing the first complete sequence of the human genome. Number 2, for the medical diagnostic, detect genetic mutation responsible for hereditary disease, allowing for precise diagnosis and personalized treatment plans. Used in prenatal testing to identify genetic abnormalities. Despite of that, it is also applied in evolutionary biology, as in constructing phylogenetic trees by comparing DNA sequences across different species to understand evolutionary relationships. And lastly, for forensic science, assists in the identification of individuals based on their unique genetic profiles in criminal investigation and paternity testing.
Mechanism of gel electrophoresis. Polycarbonate gel is important to determine size of pores. Heat also important in DNA fragment. High voltage impairment can make the migration process become faster, but heat also can distorting the separation when heat increase. Frederick Sanger method also has an advantage during the experiment, like high resolution fragment and accuracy of the fragment, because high resolution provides an ability to polycarbonate to resolve a DNA fragment. However, accuracy provides a reliable data and consistent result during the experiment, but slightly different variation can interrupt the DNA code.